the time of the COVID-19 outbreak, we immediately made the connection with the llama-derived antibody we had in our freezer because we thought it might work against this COVID-19 virus. So the next thing to do was to prove this in the lab in experiments, and we could demonstrate that our antibody binds to the new virus and furthermore can neutralize that virus.
So based on our experience with the SARS-CoV-1 virus, we knew that our antibodies can prevent the infection of that virus by binding to its spike protein and thereby preventing the attachment of the virus to its host cell. So the first thing that we did with the SARS-CoV-2 virus was to test whether our antibodies could actually do the same thing. And in a few very easy assays we could demonstrate that this was indeed the case. So next we checked whether this was sufficient to prevent also the infection of the virus by using a neutralization assay. And that's something very exciting. We were 3 people looking together through the microscope, almost fighting to see what is happening. And we were very lucky to see that one of our antibodies was also able to prevent the infection of the SARS-CoV-2 virus.
So Discovery Sciences came aboard to help to translate the antibody from the camel towards the human. So if you want to prepare an antibody for the clinic, it needs to be humanized and optimized in certain characteristics. So this is actually where Discovery Sciences brings a lot of expertise. We also helped with the animal studies. So in this case, hamsters are very susceptible to the disease, and we helped with designing the studies, and we showed actually that we can cure hamsters from COVID-19.
So at VIB Discovery Sciences, we helped out with further improving the stability properties of the antibody. So as the antibodies come straight out of the camelids, it still needs a lot of, let's say, properties that need to be improved to make it, let's say, a workable drug. So we were heavily involved in further designing the antibody, as well as testing different stability properties to make it ready to go into development.
And then we transferred it in April to UCB, which is a big Belgian biopharma company who have the necessary expertise to really manufacture drugs and then bring them to clinical trials. In the meantime, the business team of VIB contacted the necessary investors and found management, experienced management to run the company, because we figured that that would be the best way, best guarantee that the drug would really get developed, rather than perhaps licensing it to another company who may or may not develop it further. And so everything came together, and by about July the company was established, and now we're of course still helping them.
So an antibody-based approach compared to a vaccine are complementary actually. So the vaccine against COVID-19 requires your immune system to react against the vaccine, which takes some time. Whereas with an antibody therapy or antibody prophylaxis even, you have immediate protection. The antibodies that otherwise a vaccine should induce in your body are provided passively and immediately provide protection.
So in first instance we want to treat people who are really ill, who are already hospitalized, to reduce the viral load in these patients so that their immune system can take over the fight and complete the fight against the virus. In a second take, we want to develop a solution for people who have been in contact with an infected person but who may not have the disease themselves, just to reduce the chance that they will develop the disease or transmit the virus to other people.